Hello student, good morning. To continue the lecture, that is the life cycle of Echinococcus granulosus. Reproductive system. Echinococcus is hermaphrodite. The male and female reproductive organs are present in the second proglottids or mature proglottids. Male reproductive system. In the middle region of the proglottids, encircling the other reproductive organs, are about 20 to 30 spherical structure known as testes. From each testis, a fine duct arises known as vas efferens. In the postmedial portion of the proglottids, the vasa efferentia join to the base of vas deferens. Vas deferens is an extremely convoluted sperm-filled tube which passes lateral to an enlarged coiled marginal serous or penis. The serous lies in a bulbous serous pouch or sac. The serous during copulation is protruded through a lateral genital atrium and genital pore. Female reproductive system. Opening into the genital atrium closely behind the serous is a female copulatory tube, the vagina. The vaginal opening is guarded by a muscular sphincter. Behind the sphincter, the vagina passes downwards and enlarges at its inner end to form a sperm-filled sac. The seminal receptacle on the posterior side of the proglottids. There is a bilobed ovary with a narrow central isthmus from which an oviduct arises posteriorly. The oviduct joins a duct from the seminal receptacle, and this combined fertilization duct continues posteriorly into a median compact ootype. The ootype is surrounded by the unicellular Mealy's glands. In the posterior most regions of the lies a bilobed vitelline glands. The two lobes of vitelline glands are oriented dorsoventrally from the vitelline glands. Medium vitelline ducts arises the join the ootype. From the ootype, a tube-like uterus arises, which extends to the point near the anterior border of the proglottids. Eggs. Eggs resembling those of ctenoid are monoperculated and measuring about 30 by 38 micron. These are ectolithal, that is, the zygote is surrounded by the yolk cells provided by vitelline glands and cell materials. After an egg shell is laid down, the eggs pass from the ootype into the uterus. When the uterus of gravid proglottids burst, the eggs are liberated and passes out along with the feces of dog. These eggs gain entrance into a intermediate host, man or other mammals, either through water or in forage. Each egg contains a fully developed oncosphere. Human infection develops from too intimate an association with dogs. Infection of children is commonly established when dogs are permitted to lick the mouths. Eggs can be ingested from contaminated fingers. The eggs, upon being swallowed by mammalian intermediate host, hatch a duodenum as the typical 6-hooked oncospheres. The hatching occurs in the slightly alkaline medium. Activations of embryo takes place in the either human bile or intestinal juice of ruminants. They are not activated in bile or intestinal juices of dog and cats. These escaping oncospheres penetrate the intestinal wall, enters the mesenteric venules and become lodged in the capillary beds of various organs. The hooks are now said after 3 hours of egestion. The embryo reaches its final place of development among mouse, deer, etc. They reach lungs, whereas in the man and other mammals they reach in the liver, lungs, brain, kidney, spleen, etc. But liver being the most common site, implantation and growth occurs here. Oncosphere now develops into hydatid cyst. The cyst formation is slow process with the host lying down and envelopes on connective tissues around the parasite. In about 4 to 5 months, the cyst reaches approximately 10 millimeter in diameter. It goes on increasing in size and eventually becomes as big as the orange or even sometimes a football. The various types of larval cysts formed in the life history of the Echinococcus are: unilocular hydatid cyst. The hydatid cyst is composed of 3 layers. The innermost layer is called endocyst. It is thin in syncytium, forming the germinal layer. The middle layer is called ectocyst. It is made up of cuticles, which is amorphous and laminated. The outer layer is pericyst. It is composed of connective tissue and fiber secreted by the host. The cyst produces daughter cyst upon the invagination of its wall. This daughter cyst projects into the lumen of mother cyst. The cavities are filled with the sterile fluid known as hydatid fluid. Within some daughter cysts, tertiary or granddaughter cysts are formed by invagination. These are endogenous or exogenous cysts or brood capsules. The endogenous cyst falls free into the hydatid fluid of mother cyst, while the exogenous cyst, after detaching, migrates to other part of the body where they continue to grow further. The daughter cysts have fibrous cyst wall internally lined with endocyst. Rounded and hollow brood capsules are also continuous budded from the endocyst of mother cyst. These capsules remain attached for sometimes by means of narrow pedicle from the wall of these brood capsules. Minute scoliosis are formed. Scolex formation may occur from the invagination of walls either of daughter or granddaughter cyst. The anterior end is interned to form a central canal into which the form suckers opens in and into the expanded base of which the hooklets projects. The body of each scolex is filled with mesenchymal cells aggregated from the subcutaneous layer and to form the compact cup-shaped mass below the hooklets. The syncytium of the endocyst retains its embryonic potential and new brood capsules may be produced independently. Hydatids thus may expand to enormous size and containing nearly 50 liters of fluids, while thousands of scoliosis. The presence of the produces may be several damage adjacent host organs. When the infective brood capsules ingested by the final host is scolex quickly invaginate and attach to the duodenal mucosa for the first 4 to 5 days of the host. They undergoes little grow by the 14th day. The scolex has grown in size of first proglottids has appeared, the genital organs enlarge, and second proglottis appears by the 18th day. Grow, continue, and fully develop cell embryo and form the 35th day, and the life cycle begins again. Multilocular cyst. It is characterized by the many small bladders embedded a continuous enclosing measures membranes. These are found in cattle. It is considered to be results of different reaction inside the host. Alveolar hydatid in man. It is characterized by a malignant type of growth with jelly-filled proglottid vesicles embedded in a common dense stroma. The very thin laminal membranes does not restrict the germinal membranes that grows out into the surrounded host tissue. It is commonly sterile and undergoes degeneration and classification. Multivesicular pathogenesis. Bladders or cysts are harmful when they develop rapidly. They cause inflammation reaction in the surrounding tissues of the host, results in the formation of fibrous tissue. The escaping hydatid fluid not only causing diarrhea, abdominal pain, etc., but also death of the host sometimes. Treatment. Surgery is the recommended treatment for the hydatid disease, but removal of the hydatid cyst may be approached with care, and in harboring the hydatid often develops hypersensitivity to fluid. The cyst is broken during the surgery and fluid takes into the body cavity. The patient may be within the minutes from the anaphylactic shock. If the patient survive photoscolysis, which are liberated from the breakage of the cyst, may set up the second infection. In order to check the secondary infection, sometimes recommended to inject the cyst with alcohol or iodine kill photoscolysis. Mebendazole has been successfully used. Some individuals who have hydatid loculate the inoperable adult worms dogs are susceptible or treatment with praziquantel. The following are the elements of the controls prognosis of hydatid cyst. Control measures. Treatment of the infected dogs with effective anthelmic or to kill them. Keeping the viscera of domestic animals from being eaten by the dogs. Children should not be allowed to play dog frequently. Avoiding play with dogs, and hands should be thoroughly washed before the eating. This is the life cycle of pathogenicity, treatment and control measures of Echinococcus granulosus. Okay, thank you.